All right, let's jump straight into this explainer. Think of the human body as a highly complex, beautifully organized kitchen. Today, we're looking at the ultimate master recipe, the recipe of life itself. We're talking about biomolecules. These are those fundamental ingredients that interact and literally constitute the molecular logic of all life processes. It's a giant puzzle, and we're about to piece it together. I absolutely love starting with this profound paradox. I mean, think about it. A living system grows, sustains, and reproduces itself, right? But the crazy thing is, it's composed entirely of non-living atoms and molecules. So how on earth do completely non-living chemical components combine to create a living, breathing, thinking system? Well, figuring that out is the whole point of biochemistry. To get to the bottom of that, we're going to systematically move through the four main macromolecular ingredients in our biological pantry: carbohydrates, proteins, enzymes and vitamins, and finally, nucleic acids. Section 1. Carbohydrates fueling the body. We'll kick things off here, as these act as the primary energy source for us. Whenever you grab a slice of bread or some potatoes, you're basically consuming these essential little fuel packets. Now, our understanding of these molecules has actually evolved quite a bit. Historically, scientists thought they were strictly hydrates of carbon. And hey, things like glucose fit that general formula perfectly. But then, exceptions like acetic acid totally broke that rule. So today, chemists are a bit more precise. We define carbohydrates by their functional groups. Specifically, they are optically active polyhydroxyaldehydes or ketones. Quite a mouthful, but absolutely crucial. What's fascinating is how carbohydrates scale up in complexity. We basically classify them based on how they behave when broken down with water. You've got your monosaccharides. These are the simplest units, like glucose, that just can't be broken down any further. Next up are disaccharides, like your common table sugar, sucrose, which yields 2 of those simple units. And then we scale all the way up to polysaccharides. These are massive chains. In plants, this is cellulose, building plant cell walls. In us animals, we store our carb fuel as glycogen. You'll often hear it called animal starch, safely stored in your liver, muscles, and brain for the exact moment your body urgently needs a glucose hit. So the real question is, how are these massive chains actually built? Nature snaps these smaller monosaccharides together by removing a water molecule to form an oxide bridge. It's brilliant. We call this specific connection a glycosidic linkage. Take cellulose, for example. It's a straight-chain polysaccharide, and that linkage happens specifically between carbon 1 of one glucose unit and carbon 4 of the next one down the line. Section 2. Proteins, the building blocks. With our fuel sorted, let's move right along to the fundamental building blocks that literally give our biosystems structure, form, and function. So, if carbs are linked by glycosidic bonds, proteins are essentially long chains of alpha amino acids held together by peptide bonds. And amino acids, they are incredibly unique. They pack both an acidic carboxyl group and a basic amino group. Because of this, they exhibit amphoteric behavior. What that means is, in water, the carboxyl group can actually lose a proton, and the amino group can just snap it right up, forming a dipolar ion. We call this a Zwitter ion. This dual Jekyll and Hyde nature is exactly what lets proteins interact in such amazingly versatile ways inside you. We can neatly divide proteins into 2 major molecular shapes based on how those chains organize themselves. On one hand, you've got fibrous proteins. These are polypeptide chains running parallel to each other, locked tightly by hydrogen and disulfide bonds. They form these tough, insoluble fibers. Think of the keratin making up your hair, wool, silk, or the myosin flexing in your muscles. On the other hand, we have globular proteins. Here, the chains coil around to form a spherical, folded shape. These guys are usually water-soluble. Insulin and albumins are classic examples. It's really amazing to see how this builds up in structural complexity. We actually study protein structure at 4 distinct levels. First, you start with the primary structure. That's just the specific sequence of amino acids, kind of like letters in a sentence. Then comes the secondary structure, where that chain starts to coil or fold. Next is the tertiary structure, which is the overall 3D folding of that coiled chain. And finally, some, not all, but some, proteins have a quaternary structure. That's where multiple separate folded chains arrange themselves together in space to form one massive, highly functional unit. Let's zoom in on step 2 for a second, the secondary structure. We commonly see 2 main shapes here. First, there's the alpha helix, where the chain twists into a right-handed screw held firmly in place by hydrogen bonds between the turns. And second, we have the beta-pleated sheet. Imagine all the peptide chains stretched out to near maximum extension and laid side by side. Honestly, it looks exactly like the folds of pleated drapery. Now, I want you to think about this classic classroom question for a moment. When you boil an egg, the inside obviously goes from a liquid to a solid. But where does the water present in the raw egg actually go? The answer is a process called denaturation. The water, it doesn't actually go anywhere. Instead, the heat acts as a physical change that completely disturbs the delicate hydrogen bonds in the egg proteins. The neatly folded globules unfold, the helices uncoil, and the protein coagulates, basically trapping the water within this newly tangled, solid network. But here's the absolute coolest part. While the secondary and tertiary structures are totally destroyed, meaning it loses all biological activity, the primary structure, that fundamental sequence of amino acids, remains perfectly intact. No way, right? Moving right along to Section 3, enzymes and essential vitamins. These are the highly specialized workers of our biological kitchen. Think about digestion, absorption, energy production. These are complex sequences of chemical reactions, and they need to happen incredibly fast, but under the very mild temperature and pH conditions of our bodies. This entire operation is only possible thanks to enzymes. They're specialized biocatalysts. Almost all enzymes are globular proteins, and they are incredibly, almost unimaginably specific. Like one enzyme usually only has one single job for one single substrate. For instance, the enzyme maltase, its sole purpose in life is to catalyze the hydrolysis of maltose into glucose. It literally won't work on anything else. Alongside those enzymes, we have vitamins. These are organic compounds we need in small amounts to perform specific biological functions. We classify them by solubility. Fat-soluble vitamins, that's A, D, E, and K, can be safely stored away in your liver and fat tissues. Water-soluble vitamins, on the other hand, specifically the B group and vitamin C, well, they can't be stored in the body because they're readily excreted in urine. The absolute most critical takeaway here, you have to constantly replenish those water-soluble vitamins through your daily diet. They won't just stick around. Finally, Section 4, Nucleic Acids, Genetic Blueprints. We've reached the ultimate instruction manual for the entire biological recipe. These are the molecules completely responsible for heredity. Nucleic acids are essentially these massive polymers made of units called nucleotides. A single nucleotide is built from three things: a base, a pentose sugar, and a phosphate group. To build the long chain of a nucleic acid, these nucleotides have to join together. That connection happens very specifically between the 5' and 3' carbon atoms of the pentose sugars, creating what we call a phosphodiester linkage. There are two main types of nucleic acids controlling our biological destiny: DNA and RNA. And it's super important to note their exact chemical differences. Structurally, DNA is a double strand, while RNA is generally a single strand. Chemically, DNA uses a sugar called 2-deoxyribose, whereas RNA just uses normal ribose. Plus, while both share the bases adenine, guanine, and cytosine, DNA utilizes a fourth base called thymine. RNA actually ditches the thymine and uses a base called uracil instead. Back in 1953, James Watson and Francis Crick proposed the now iconic double-strand helix structure for DNA. It's this elegantly simple model that resembles a gently twisted ladder. The rails of the ladder are made of alternating sugar and phosphate units, and the rungs, those are the pairs of bases. Because of their specific chemical shapes, these two strands are perfectly complementary. Adenine always, and I mean always, forms hydrogen bonds specifically with thymine, and cytosine always bonds with guanine. This elegant, highly specific base pairing explains exactly how DNA can duplicate itself during cell division. It's the sole chemical basis of heredity. The exact message for synthesizing every single protein in your body is coded right here. This single molecule is exclusively responsible for maintaining the identity of every different species on Earth over millions and millions of years. It's honestly mind-blowing. As we wrap up this explainer, I want to leave you with this thought. We've spent our time breaking down these incredible, non-living chemical ingredients, the carbs, the proteins, the enzymes, and the nucleic acids that perfectly coordinate to create life. But today, our understanding goes way beyond just observation. We now possess recombinant DNA techniques. We can actively cut, paste, and alter this master code. So, since we now understand the chemical ingredients of life so intimately, how will our ability to consciously rewrite this biological recipe change the world of tomorrow? Something to think about.